Good afternoon, everyone. Yesterday we discussed enzyme properties. What are the factors affecting the enzyme action? What is the rate of enzyme action? What is the impact of temperature on enzyme action? Impact of pH on enzyme action? Substrate concentration impact on enzyme action. In last class we discussed. Today there is holo enzymes, simple enzymes. On the basis of composition in the enzymes, here two types of enzymes are there. Those are the simple enzymes, holo enzymes. Generally, what I told here, all enzymes are made up of proteins. Only ribozyme is exception. Except the ribozyme enzyme, how these almost all enzymes are made up of proteins. So that here enzymes are made up of protein. If the enzyme is made up of protein, such enzyme of it will be called simple enzymes. If enzymes are made with proteins, such enzymes are called as the simple enzymes. So protein made enzymes, how they can be called simple enzymes. Here holo enzymes. Holo enzymes, they are also called as the conjugated enzymes. Complex enzymes or conjugated enzymes or holo enzymes. What is meant by holo enzymes here? They are also called as the conjugated enzymes. Holo enzymes are also called as the conjugated enzymes. What is meant by conjugated enzymes? In these enzymes, two parts are there. Protein part is there, non-protein part is there. This P for protein part, here NP for non-protein part. In conjugated enzymes or holo enzymes, there are two parts. One is the protein part, another one is the non-protein part. The protein part of holo enzyme is called as apoenzyme. One more question, is the important one. What is apoenzyme? What is meant by apoenzyme here? The protein part of holo enzymes is called as the apoenzyme. What is meant by apoenzyme? Protein part of holo enzymes. In holo enzyme, two parts: protein part, non-protein part. That means other than protein. Protein part of holo enzyme is called as the apoenzyme. Non-protein part of it will be called cofactors. Non-protein part of holo enzyme is called as the cofactor. These cofactors may be coenzymes, prosthetic groups, metallic cofactors. These cofactors may be coenzymes or prosthetic groups or another one metallic cofactors. What is meant by coenzyme? In this non-protein part made with organic composition. This non-protein part, how this is made here, made with organic composition or inorganic composition. Non-protein part may be made of organic composition or inorganic composition. If it is made of organic composition, it can loosely bind to this here protein part, can be called as this here coenzyme. You see, this is a holo enzyme. This is a holo enzyme. How many parts are there in holo enzyme? Protein part is there and non-protein part is there. Two parts. Protein part and non-protein part. But non-protein part, how it is made? Organic composition or metallic inorganic composition. Non-protein part may be organic or inorganic. Two cases, organic or inorganic. But organic, again this organic composition, how it will be bind to protein? Loose association, tight association. Non-protein part is made with organic composition. It may loosely bind into protein part or apoenzyme or tightly bind to this here apoenzyme. If it organic composition loosely bind, that can be called as coenzyme. Tightly bind, that can be called as the prosthetic group. If it is inorganic composition, that can be called as the metallic cofactors. You see, we comes to the holo enzymes here. This is holo enzyme. Here, this is the holo enzyme. The holo enzyme, what would be there? Protein part is there. And what should be there here? Non-protein part is there. This is what is this enzyme? Holo enzyme. Holo enzyme consists of two parts. Protein part of holo enzyme, how it will be called here? Apoenzyme. What is apoenzyme? The protein part of holo enzyme is called as the apoenzyme. Non-protein part, how this is called here? Non-protein part here. There may be a coenzyme, it may be organic composition. Non-protein part, it is made up of organic composition or inorganic composition. Non-protein part of all enzyme may be organic or inorganic. But these organic composition loosely binding. It may be how it will be loosely binding or tightly binding. Loosely or tightly binding. Suppose here it is a loosely binding. The organic composition it can be binding. How it is binding here? Loose association. Then how it will be called loose association can be called as the coenzyme. Organic composition is tightly binding. How this is called here? Prosthetic group. What is meant by prosthetic group? If the non-protein part made with organic composition, such organic composition is tightly binding to this apoenzyme is called as the prosthetic group. What are working as a prosthetic group? NADs, FADs, they are acting as a prosthetic groups. And next one, inorganic composition. Here they are called as the metallic cofactors. How they can be called here? Metallic cofactors. In holo enzyme, there is an inorganic composition is there. Inorganic composition means zinc or magnesium, like this here manganese, etc. Here inorganic metals will be there. Carbonic anhydrase. What will be present in carbonic anhydrase? Zinc is there. Here you see magnesium. Where it will be there? Magnesium. They are present in rubisco, hexokinase. Manganese. Several enzymes here involving in protein synthesis. Manganese is there. Like this here, and oxygen evolving complex. Manganese is there. Oxygen evolving complex is there. Enzyme complex involving in light reaction of photosynthesis. There, what is the cofactor? Manganese is a cofactor. Like this here in enzymes here, all our enzymes here, protein part, non-protein parts are there. Protein part is called as the here apoenzyme. Non-protein part may be organic or inorganic metals. Organic composition is there. Again, according to binding with apoenzyme, then it is divided into two parts. What is this here? May be coenzyme or prosthetic group. Coenzyme means organic composition loosely binding with apoenzyme, that is called coenzyme. Prosthetic group means here the organic composition in holoenzyme tightly bind to apoenzyme is called as the prosthetic group. The metallic cofactors means in place of organic composition, if non-protein part is occupied by the metals, magnesium, zinc, calcium, manganese, like these metals occurs in non-protein part, that can be called as the metallic cofactors. Like this here, what is this here? These are the enzymes. Simple enzymes are there, holoenzymes are there. Simple enzymes means only they are made up of proteins. Holoenzymes means protein and non-protein parts will be there. These are the, according to composition of enzymes here, simple holoenzymes will be there. These are here types of enzymes on the basis of composition. And IUB classification. Enzymes International Union of Biochemistry classified the enzymes. How it is classified here? 6 classes. Hydrolases, oxidoreductases, transferases, lyases, isomerases, ligases. Those classification of enzymes, let us see. Classification of enzymes on the basis of the enzymes catalyzing reaction. Reactions. Classification of enzymes. On the basis of enzymes catalyzing the chemical reactions. The nature of catalyzing chemical reactions here, how it will be classified here? The IUB. IUB means International Union of Biochemistry. What is the IUB? International Union of Biochemistry. International Union of Biochemistry. International Union of Biochemistry, IUB. So IUB classification, what is the IUB? Here, International Union of Biochemistry classify the enzymes into 6 classes. Total enzymes here, how many classes it is classified here? Enzymes are classified into 6 classes. What are those 6 classes here? Hydrolases. They are the hydrolases. Then oxidoreductases. Oxidoreductases, hydrolases, oxidoreductases, transferases, hydrolases, oxidoreductases, transferases, ligases, isomerases, and ligases. These are the, what are these here? 6 classes. These enzymes here, IUB, International Union of Biochemistry, classified the enzymes into 6 classes. Hydrolases, oxidoreductases, transferases, ligases, isomerases, ligases. Hydrolases, again they are divided into 2 types, 2 subclasses. What are those 2 subclasses here? Phosphatases, peptidases, oxidoreductases, here oxidases, reductases, and transferases, oxidases, reductases, transferases. Here, oxidoreductases and transferases, transaminases and kinases. Transaminases and kinases. Like this here, there is a further classification. IUB, International Union of Biochemistry, classified the enzymes into 6 classes. First class, hydrolase. Second class, oxidoreductase. Third class here, transferases. Fourth class here, ligases. Fifth class, isomerases. And sixth class, ligases. Again, in this class 1 hydrolases, there is further division into 2 subclasses, phosphatases, peptidases. Oxidoreductases divided into 2 subclasses, oxidases, reductases. Transferases is divided into transaminases, kinases. Here there is no subclasses in the ligases, isomerases, ligases. There would be, what would be there? 6 classes of enzymes are there. So what is this hydrolases? They catalyze the reaction in the presence of water. The enzymes which are catalyzing the reaction in the presence of water, such enzymes, how they can be called here? Hydrolases. But these hydrolases are how many types here? 2 subclasses. Phosphatase, peptidase. What is phosphatase? What is peptidase? We see. First one is the hydrolases. Class 1 here, hydrolases. Class 1 is the hydrolases. These hydrolases are divided into 2 subclasses. What are those here? Subclass 1, phosphatase. What is mean by phosphatases here? In the presence of water, the enzymes which can catalyze the reaction for removal of phosphate from substance in the presence of water, those enzymes are called as the phosphatases. What is mean by phosphatases here? The enzymes here. Suppose you see here fructose 1,6 biphosphate. You see this is a substance. Fructose 1,6 biphosphate. Here fructose 1,6 biphosphate. What is this substance? Here 1,6 biphosphate means It is a fructose sugar. There is 6 carbons are there. First carbon position phosphate is there. Sixth carbon position phosphate is there. That meaning here 2 positions phosphate is there. Therefore, it is called biphosphate. What is the position of phosphate? First carbon, sixth carbon. Therefore, it can be called as the fructose 1,6 biphosphate. But this is here converted into fructose 6 phosphate. How this is converted? Fructose 6 phosphate. You see, this is fructose 1,6 biphosphate converted into fructose 6 phosphate. What is it meaning? First position phosphate is lost. So here, this removal of phosphate, what is the enzymes involving here? Phosphatases. The enzymes which involves in the removal of phosphate group from substance that can be called as this here phosphatase enzymes. Suppose here it is a substance, there is a 2 phosphates will be there. Here phosphate and phosphate. S for substance. What is this here? This is the S. S for what is this S? Substance. This substance has 2 phosphates. First position, last position. Here what is working? The enzyme is working. What is the enzyme? Phosphatase enzyme is working. Phosphatase enzyme, what it is doing here? Removing the phosphate. This inorganic phosphate, it has been removed. Then what would remains here? Substance plus phosphate remains. Out of 2 phosphates, 1 removed. But to remove a phosphate from substance, what is the enzyme involves here? Phosphatase involves. But example for what is this here? Fructose 1,6 biphosphate means phosphofructophosphatase. It will be called as the phosphofructophosphatase here. Whatever this is from substance phosphate is removed, that can be called as here phosphatases here. Here pseudoheptalose diphosphate. From this phosphate is removed, phosphatases. So anyway, from substance phosphate is removed, phosphatase is working. But here, what is the medium? Water. It is done in the presence of water. Henceforth, it is called as the hydrolase. Next one, peptide is this. What is the peptide is this? Second one. A, peptide is this. What is meant by peptidases here? Peptidases are these are the enzymes which involving in the breaking of peptide bonds. With results, what will be the amino acids are formed. The presence of water, generally by removal of water, peptide bond is formed. By addition of water, peptide bond is breaking. This is a protein chain. Amino acid, amino acid, amino acid, amino acid, amino acid, protein chain. This protein chain here, what is the enzyme? Peptidase enzyme. Peptidase enzyme. Now here, what in the presence of water? We add the water. What is breaking? Dipeptide, tripeptide, tetrapeptide, pentapeptide. Peptide bonds are breaking. Which results what will be formed here? Amino acids can be formed. So the breaking of peptide bonds is done by the enzymes called as the peptidases, which results proteins are forming amino acid. So by the removal of water, peptide bond formed. By addition of water, peptide bond is breaking. The enzymes which are adding water to peptidases, therefore peptide bonds are breaking, henceforth amino acids have been formed. So the enzyme which involving in breaking of peptide bond, those are called as the peptidases, which results peptides can form amino acids. This is the phosphatase peptidases, hydrolases. Second category, what is the second class? Oxidoreductases. Another class of enzymes, oxidoreductases. Second class, oxidoreductases. First class here, hydrolases. Second class, oxidoreductases. Oxidoreductases are divided into what is here? 2 subclasses. What are those 2 subclasses? Oxidases. These are the 2 subclasses, oxidases. What is meant by oxidases here? The substance is added with oxygen, oxidase. Here, what is the oxidases here? you see any substance, the substance is added with this oxygen. Here glycolic acid. Glycolic acid. This is by adding oxygen. Glyoxolic acid. Glycolic acid, glyoxolic acid. So what is the enzyme? Oxidase. What is the oxidase here? The enzymes which involving in the addition of oxygen to substance, such enzyme is called as the oxidase enzyme. So here addition of oxygen induced by enzymes called as the oxidases. And here second one, reductases. What is the reductases here? These reductases. Oxidases, reductases will be there. What is meant by reductases here? Here addition of oxygen, oxidase. But reductase means removal of oxygen is a reductase. Or removal of hydrogen. Here reductase. But reductase mostly removal of oxygen only helping. Another category dehydrogenase is there. That is removal of hydrogen. Reductase is two categories again. What is it? Directly removing the oxygen reduction. Suppose you see NO3, nitrite. Nitrite is converted into nitrate. Here, what is the enzyme working? Nitrite reductase. Nitrite reductase. Nitrite reductase. Here, nitrite reductase means what is the nitrite reductase here? The enzyme which involving in the removal of oxygen from nitrites. Those enzymes can be called as the reductases. If here NO3 is converted into NO2, one oxygen is removed. Removal of oxygen, which will be called reduction. So here, this is from where it is removed oxygen, nitrite. Henceforth, it will be called nitrite reductase. Reductases involving in the removal of oxygen from substance. Those are called reductases. And one more subclass is there in oxidoreductase, you see, that is also reduction, but it is dehydrogenation. What is meant by dehydrogenation? From substance, it removes hydrogen. Malic acid is there. Here, malic acid is converted into, how it will be called oxaloacetic acid. Any substance you take, from that substance, what would be there? Hydrogen is removed. Suppose here, this is a substance. From this substance here, what would be there here? This hydrogen is removed. But it is added to, suppose it is substance plus hydrogen, NAD plus it is. Then this removal hydrogen, where it is adding NAD plus. So that here NAD is reduced into NADH. Then what would be there? Substance is formed. Therefore, here the removal of hydrogens from the substance. This removal hydrogen added to NAD places, FAD places. So in the cytoplasm, NADHs, FADHs can be formed. Therefore, here what is here? Oxidoreductases, maybe oxidases, maybe reductases, maybe dehydrogenases. Like this here in second class, oxidoreductases here, three subclasses will be there. And one more thing, what would be there? Hydrolysis, oxidoreductases, transferases. Transferases. Third one, transferases. Third class, what is this here? This is a transferases. What are the transferases here? These are the two types, transaminases and kinases. Transaminases and kinases. What is meant by transaminases? They can, suppose here, this is the two products will be there. It is a reactants here, substance is there, amine group is there. Here plus it is a substance is there. But here next what would be there? It is a substance A, this is a substance B. Here the enzymes, what is the enzyme? Transaminase. What is meant by transaminase here? The enzyme which involving in the transfer of amine group from one substance to another substance. Such enzymes can be called as the transaminases. Actually, amine where it is there, at the A substance amine is there. But A2 where it is adding to the B. So that here next by action of the transaminase, B is getting amine group, A is free from amine. Otherwise, here two amines are there. One amine is donated to here. So that what is this here? Simply transaminases, they are aminases or enzymes. They can transfer amine group from one substance to another substance. Such enzymes are called as the transaminases. Example, aspartic acid is there, alpha beta-glutaric acid is there. Suppose here, for example, A means, what is the A? You take a aspartic acid. One example, aspartic acid. In place of B, alpha beta-glutaric acid you can take. Here, aspartic acid, alpha beta-glutaric acid. But from this aspartic acid, what is transferred to alpha beta-glutaric acid? Amine group. Which results, what would be formed? Alpha beta-glutaric acid. It will be converted into glutamine. Then how it is there? Glutamine has been formed. Then alpha aspartic acid, how it is converted? Oxaloacetic acid. Here, this aspartic acid transfer amine group to alpha KGA. Therefore, alpha KGA converted into glutamine. Then, as it is donating amine, aspartic acid it will become a oxaloacetic acid. Simple thing, the aminases can transfer amine group from one substance to another substance. They can be called as the aminases. So here, transferases two types: transferases and transaminases and kinases. Then second one, let us discuss the kinase. What is the kinase here? They can transfer phosphate group from substance to ADP. Suppose here 1,3,5 is here. Phosphoenolpyruvic acid, ADP. It will become pyruvic acid. Then here, what is working? Kinase is working. You see, this is a substance. What is this? Phosphoenolpyruvic acid. It is adenine diphosphate. Here in this substance, phosphate is there. In this substance, phosphate. How many phosphates are there? Two phosphates. Here, adenosine diphosphate. Suppose it is adenosine. Phosphate, phosphate. How many phosphates are there? Two phosphates. Then how it will be called? Adenosine diphosphate. This is some substance, phosphoenol. Here it is a pyruvic acid. So phosphate group is there in the phosphoenolpyruvic acid. Phosphate is there. This phosphate, where it is transferred, here it will be transferred. Therefore, here this is ADP, how it is converted? Adenosine triphosphate. So the enzyme which is involving in the transfer of phosphate group from one substance to another substance, that can be called as the kinase. Then how it will be converted? Pyruvic acid. So here the transfer of phosphate from one substance to another substance is induced by enzyme called as the kinases here. Kinases can transfer phosphate from one substance to another substance. Transaminase can transfer amine group from one substance to another substance. Like the transferases are transaminases and kinases, there are two subclasses will be there in the transferases enzymes. So hydrolysis, oxidoreductases, transferases. Next one, lyases. And lyases, isomerases, lyases. Lyases, first one. Fourth class here, lyase. Another, there are 6 classes of enzyme. Class 4, lyase enzymes. Lyases. Here, what is the meaning of lyase? You know that in the name only, meaning is there. Lyases means lysis. The enzymes which are involving in the breaking of substance or breaking of bonds, which results here, different products will come here. Suppose arginosuccinic acid, it can be broken by lyase enzyme, which results succinic acid, fumaric acid can be formed. This is a arginosuccinic acid. What is this substance here? Arginosuccinic acid. This arginosuccinic acid enzyme, what is the enzyme? Lyase enzyme. Which results, what would be there? Succinic acid can be formed, and here fumaric acid can be formed. So succinic acid and fumaric acid, by this breaking of arginosuccinic acid, succinic and fumaric acids can be formed. So here the enzymes which are involving in breaking of substances into different products, such enzymes can be called as the lyase enzyme. Lyases are nothing but here involving in breaking of substance into different products. That is the lyases. Next one, isomerases. Class 5. What is the here class 5 enzyme? Isomerases. What is mean by isomerases here? The enzymes which involving in the interconversion of isomers from one form to the another form. Suppose you see glucose 6-phosphate, fructose 6-phosphate. Glucose, fructose, what are these? Isomers. Here it is a glucose 6-phosphate. Isomer is glucose 6-phosphate is there. This is the what is this here? Glucose 6-phosphate. And next one, fructose 6-phosphate. Here, what are these here? Fructose 6-phosphate. Glucose 6-phosphate, fructose 6-phosphate, both are the isomers, but they are interconverted each other. Ribose phosphate, ribulose phosphate. RB means ribose phosphate. RUB means ribulose phosphate. Ribose phosphate, ribulose phosphate. What are these here? Isomers. So isomer means the molecules which have same molecular formula but having different structure. Structurally they are different, formula is same. They are these glucose 6-phosphate and fructose 6-phosphate. They are the isomers. In the presence of isomerase enzyme, they are interconverted each other. The enzymes which involves in the interconversion of isomers from one form to another form, such enzymes are called as the isomerases. And RP and RUP means ribose phosphate, ribulose phosphate. Here is also what is this enzyme? Isomerase. Here it is a phosphofructo isomerase or exoisomerase it is called, because here according to substance this isomerase is called. It is a ribose isomerase. It is exoisomerase. Like this here we will call isomerase. Chemical nature is isomerase only. But here in front of chemical nature of enzyme we add the substance name. Exoisomerase, riboisomerase. For distinguish this here isomerase to isomerase, we will add the substance name. Like this here, these are the isomerase enzymes. And the last class enzymes, what would be there? There is the ligases. Here, ligases, isomerases, ligases. Ligation, you know in what ligation means? Binding here. So here, the addition of molecules to the substance, the enzyme which catalyze the reaction in which the one substance is added with the different molecules. Addition of molecules to substance is induced by enzyme called as the ligase enzyme. Suppose here glutamic acid is added with ammonia, then here ATP. What would be formed? Glutamine. Glutamic acid will become amine because here ammonia is added to glutamic acid. You see this here, that one, the enzyme isomerase, then last one ligases. Class 6, what is this enzyme? This is the ligases. What are ligases here? The enzymes which involving in the addition of molecules to the substance. Here the substance, what is the substance here? Glutamic acid. This is a substance. Glutamic acid is added by what is ammonia. In this case, what is required? Energy, ATP is required. Now what will be formed here? This addition of molecules to the substance. Glutamic acid, how it will be converted? Glutamine. Glutamic acid is converted into glutamine. Then here, what would be there? ADP plus inorganic phosphate can be formed. So the addition of ammonia to the substance, it will be called here ligase enzyme. The enzyme which involving in the addition of molecule to substances can be called ligases. Here ammonia is added to glutamic acid, therefore glutamic acid can be converted into glutamine. This is what is here. There is the different classes of enzymes. Hydrolases, oxidoreductases, transferases, ligases, isomerases, ligases. 6 classes of enzymes are there. Hydrolases, there are again divided into 2 subclasses here. What are those here? 2 subclasses. Hydrolases, phosphatases, peptidases. Oxidoreductases, oxidases, reductases, as well as dehydrogenases. And here transferases, transaminases, kinases. There is no further division in case of lyase, isomerase, ligases. Like this here, this is the enzyme classification. These enzymes, according to composition, two different types of enzymes are there. Simple enzymes, hollow enzymes. Simple enzyme means, what is meant by simple enzyme? Only it is made up of protein. The enzymes which are made up of protein only, those are called as the simple enzymes. What is meant by Holo enzymes here? They are made up of protein and other than protein parts, the non-protein parts. The enzymes which have a protein and non-protein part, such enzymes called as the Holo enzymes. In the Holo enzyme, the protein part is called as the apoenzyme. What is apoenzyme? One more question. The protein part of Holo enzyme is called as the apoenzyme. And non-protein part, how this non-protein part is made? Maybe organic or inorganic. If it is made with organic, again two subcategories. Prosthetic group, coenzyme. What is coenzyme? Non-protein part is made up of organic composition. It can be loosely bind with or loosely associate with apoenzyme, that can be called as the coenzyme. In the non-protein part made with organic composition, it would be tightly bind to apoenzyme, that can be called prosthetic group. These prosthetic groups here, normally what are acting as a prosthetic groups here? Heme is a prosthetic group. Heme is a, what is this here? Prosthetic group. NAD, FAD, these are acting as a prosthetic groups here. And now when you see this here, metallic cofactors. In the enzymes here, the non-protein part is a metals, magnesium, zinc, manganese, like these metals are there, those can be called as the metallic cofactors. Like this here, the enzymes here, simple and Holo enzymes will be there. And in this case here, IUB, International Union of Biochemistry, classified the enzymes in how many classes? 6 classes of enzymes. These are the classes of enzyme, composition of enzymes here. Then your assignment, what is this relevant with the enzymes assignment? What is Holo enzyme? What is apoenzyme? Question number 1. What is Holo enzyme? Assignment. What is apoenzyme? What is apoenzyme? What is coenzyme? What is coenzyme? What is prosthetic group? Question number 4. What is prosthetic group? Prosthetic group. Question number 5. What are transaminases? Transaminases. And what are phosphatases? What are phosphatases? What are peptidases? What are the peptidases? What are peptidases? What are peptidases? What are phosphatases? What are kinases? What are kinases? What are kinases? Phosphatase, peptidase, transferase, kinases. Here, next one, what is here? Another one. Question number 9. Ligases. What are ligases? What are ligases? What are ligases? Ligases. Ligases. Isomerases. What are isomerases? Like this here, these are the here assignment on the enzymes. What are isomerases? What are ligases? What are ligases? What is prosthetic group? What is coenzyme? What is apoenzyme? What is Holo enzyme? And simply one more thing, the last question also you write some cases here. What is simple enzyme? Simple enzyme. And another one, this is important one. Question number 30. What is ribozyme? What is ribozyme? Competitive point of view, here it is an important one. Ribozyme. Ribozyme. What is meant by ribozyme? How it is made here? So this is a competitive point of view. Ribozyme is made up of normally proteins here involving in formation of enzymes here. All enzymes are made up of proteins only, but exception is there. Where is that ribosome? How this is made? RNA. What kind of RNA? 23 is rRNA. Ribosome, it is an enzyme made up of RNA. It is made up of 23 is rRNA working as a catalyst. So this is an enzymes assignment. And tomorrow what we have to discuss here, then factors affecting the enzymes, competitive inhibition, non-competitive inhibition, allosteric modulation will be there. Then one more thing will be there, the potential energy, activation energy. Without activation energy, how these products will be formed. With activation energy, how it will be there. That's one thing is there. One graph here, activation energy and progression of reaction. And next one, factors influence this here enzyme action. Those are the competitive inhibitor, non-competitive inhibitor, allosteric modulation. These are your biomolecules information and living state as a dynamic state. Next class tomorrow we will discuss. Okay, thank you one and all.